all the long way from Oxford. 4 trains though, apparently. Yeah, she's got a lot. 4 trains to get here from Oxford. Who does infectious disease and general medicine at Oxford University Hospitals, but probably more relevant today, does a lot of human genetic works around almost every topic as far as I can tell, related to antibodies and infection. He's going to talk about antibodies, but he's got a wide range of interests. I'm sure you can ask him almost anything and he'll answer. That's not pretty cool.
Thank you very much for the introduction, Gus, and thanks very much for the invite. It's great to come here and see the hospitals and see where you do work, and I can see quite a few people online as well, so thanks very much. Yeah, as Gus mentions, yes, I'm an infectious disease consultant that got a small lab in Oxford, and our main focus is trying to understand infections in more detail. So this is what really stimulates us and what makes us work. So 2 opposite ends of the spectrum. On the left is a patient, looks like they're sleeping, they're not, they're critically unwell, looks quite young and fit. It's got devastating community-acquired pneumonia, really so unwell that he's had to be prone, so he's had to be laying on his front to ventilate because we're not able to get enough oxygen into his lungs, and he's paralyzed so that we can do that and ventilate him appropriately. And if we switched off the ventilator, he would die. And then on the other side of the coin, is the picture on the right, is seeing infections that can cause completely different things. So cancer. So this is a cancer of the skin. This is Kaposi sarcoma. It's caused by HHV8, so one of the human herpes viruses. So there's sort of diverse ways of thinking about infection and its impact, either on a sort of an acute level with patients with high mortality, and then sort of the longer term picture of causing other diseases that we may not have traditionally thought of as being caused by infections or so-called non-communicable disease. So how can we identify and treat these infections more appropriately? So there's lots of challenges. I'm sure many of you guys think about this an awful lot as well. Thinking about the patient who was on the left, the sort of critically unwell patient, discovering sort of new drugs or testing new drugs in that kind of environment is very logistically difficult because patients are coming in suddenly into hospital. You've got to support them, provide lots of life support, and so sort of testing a new drug in that kind of circumstance can be quite difficult, and working out whether it's going to be effective or not can be difficult. And actually knowing whether or not a patient even has an infection, never mind which infection, can be very difficult. So actually looking at this patient group is quite difficult generally in terms of studies. Then if we're thinking about the patient more on the right, from the previous picture, thinking about linking infections to possible other non-communicable disease, often we think infections, if you're going to be exposed, would be quite a long time before patients develop these types of diseases, be they cancer or cardiovascular disease or some other non-communicable disease. So identifying that link and providing evidence that there's a link between infection exposure and sort of non-communicable disease is quite difficult. We and others have tried to use genetics for many, many years to try and understand these links and understand what might be putting people at risk and how we can identify infections better and maybe treat infections better. But our Manhattans for many years have looked very much like that at the top left, very flat with nothing really to show, despite a reasonable number of samples of a variety of infections that we've looked at. And that's when we're really thinking about infections as a case and a clinical definition of an infection, and then sort of population controls, people that don't, and we don't think have ever had an infection. So it was all quite difficult until obviously COVID came along, and obviously although COVID was difficult for many people from an infection looking at genetics perspective, it was quite helpful because it does tell us if you've got a large enough sample size in terms of people that you can identify as cases, and then maybe controls that you can look at in some way, you've got a distinct infection that you can classify specifically in an individual with a clinical syndrome that fits with sort of COVID, then that is very helpful. And then when you do that, you can clearly see a number of genetic association signals that generally make sense. And even better, it's difficult to say that the genetic associations were what drove new sort of drug discovery, but they certainly help contribute to it. And a genetic variant at TIC2 that highlighted certainly helped prioritize a specific drug to be used in COVID. That was baricitinib. And although it came late in the pandemic, I've used it quite a lot for patients, and it does work in certain subgroups of patients. It can be an incredibly effective drug. So although COVID was difficult, this was really helpful because it says that if you do look hard enough, actually genetics can be helpful and informative. And a lot of these genetic association signals are still very potentially meaningful and worth a lot of exploration. And as always, it's going to take quite a long time to get to that next step. I've made a massive assumption here that I don't need to talk through Manhattan plots, I presume, but obviously do stop me if anything I say needs more justification or description. So in an ideal world, for that problem, understanding sort of drivers of infectious disease, bugs that might cause that, genetic heterogeneity that might stratify individuals' risk of developing an infectious disease, or on the other side, trying to think about identifying links between infection exposure and disease, non-communicable disease, and using genetics. It'd be great to be able to do vast studies such as we did with HGI and with the COVID GWAS, but obviously no one's going to fund that type of work for specific infections, especially if we can't diagnose them. Then you've got lots of other challenges. Well, who do you define as cases? How do you define your controls appropriately? Are they exposed, non-exposed? There's lots of questions that are risen with that. So we as a group try and use alternative approaches towards defining infection. So rather than defining cases as sort of infectious diseases episodes sort of presenting in hospital, we generally try and use immune readouts instead as sort of proxy measures of infection exposure. And then we can go another step forward. So rather than just looking at immune readouts to natural infection or wild type infection, we can also look at immune responses following vaccination, because with vaccines, we know exactly when people have been exposed to that sort of vaccine or antigen of interest, and we've got a very discrete measure of time following that exposure. And then as a group, we also try and work on trying to take those findings closer to the patient and working with patients either in hospital settings or out in the community to try and understand how we can use this type of information, mainly through genetics, to improve the outcomes of patients with infection-related disease. So I'm going to talk through a few case examples, which I'll just go into the next slide. But as Gus mentioned here today, I'm going to talk a lot about antibody measures, because that's generally what a lot of our work is focused towards. And when we talk about antibody, in this talk, I'm going to talk exclusively about immunoglobulin G. So the sort of marker that we always think of as being a likely measure of prior exposure or indeed chronic infection with a multitude of infections. We treat this as a quantitative trait or as a continuous trait, which obviously gives us statistical benefits as opposed to going for a more case control analysis. And it allows us to look on a larger number of individuals, and that we can measure do they have very little or no detectable antibody, or do they have very high levels. We do make a lot of assumptions in doing this. We make assumptions that the level that we measure in an individual at any time is pretty stable, which I'll come back to a little bit later. But the really nice thing about antibodies is we can look at lots of antibodies simultaneously, and I'll talk through some of that later. Generally, we've been working with hundreds, but there are now options to look at thousands or tens of thousands of antibodies simultaneously. So you can look at very rich data sets. From a logistical perspective, looking at antibody against sort of IgG is really great. People say, oh, what about the samples? These have been in the freezer for years. We've thawed them a couple of times. There's no problem with IgG. You can freeze and thaw samples. You can leave them on the bench side for a while. The IgG is stable. It's a great thing to work with. And as I mentioned, looking at antibodies following vaccination is really helpful because you know exactly when people are being exposed to the vaccine and can measure antibodies, which helps a lot. So if I've got time, I'll take you through 4 of our sort of case examples to hopefully persuade you that looking at antibodies can be helpful. But at the same time, tell you a little bit about some of the challenges that we've faced and are continuing to face and continue to cause me a lot of headache and pain, and probably no doubt for the future as well, just as I've sort of put these slides together. So nowadays, I usually start with this one as the first case example, because it seems a shame not to continue to talk about COVID, even though most people roll their eyes. Isn't that way gone? But actually, it was helpful academically, and it helps, gives us lessons to take forwards. So this is thinking about vaccination. So I'm going to move between thinking about antibodies following natural infection or natural exposure and vaccination challenge. And obviously, in a sense, looking at SARS-CoV-2 antibodies following vaccination was really helpful, because generally it's a very naive background. It was a great time to do that. So we can be looking at sort of naive antibody responses. So people who haven't been exposed to it previously. So very lucky to be working in Oxford at that time, and with lots of great people. Can't quite see everyone there, so let me just move that down actually. So Andy Pollard, Tess Lamb, Daniel O'Connor, Gillian Knight, fantastic collaborators. I mean, got wonderful collaborators all around the world. Very lucky to work with lots of people. For this particular project, these people were absolutely quintessential. So we were looking at the vaccine trial for the Chaddox vaccine, and Andy and Tess particularly had so much foresight that they just wrote in genetic studies into their clinical trials de facto. It was just going to be done. So individuals received the sort of Chaddox vaccine, which obviously is sort of dependent on spike protein. I've shown the molecular structure of spike protein on the right. That's what we think the immune system mounts a response against, which is probably protective, stops people getting more infected than they should do. And it's particularly antibody responses that we think are probably the most protective response. And generally, we think targeted towards the receptor binding domain, which is just there highlighted at the bottom right. So we were very lucky for lots of reasons. What I'm going to talk about now is mainly our sort of discovery angles. So that's looking at the initial trial of the Chaddox individuals. We had just over 1,000 individuals that had given their consent to have genetic studies. I mean, pretty much everyone gave their consent for genetic studies. And we had antibody levels against all of spike and all of receptor binding domain available across all individuals anyway. That was just being done as part of the clinical trial. So it made our life much easier for doing this type of analysis. So we plan to do a genome-wide association study looking at antibody responses, or the genetics affecting antibody responses at various time points following vaccination. Our discovery was focused at day 28 following the first vaccine. That's where we expected to see the greatest amount of variance following the first stimulation. That's exactly what we saw. And also that's where we'd expect to have the most endpoints, because there was still a lot of enthusiasm at the early stage of the clinical trial, and we'd have less loss to follow-up. We just sort of treated this as a sort of normally distributed trait, which it was anyway. We just normalized it a little bit more. And we ran our GWAS, and we saw immediately an association across chromosome 6, which, unsurprising, if you think about infection traits, is very much, is that going to go down there? Yeah, hopefully that will work, is localized in the class 2 MHC region, falling on HLA, and particularly HLA-DQ genes. I'm not going to spend too much time talking about HLA in this talk, because usually when I do, I get lots of people falling asleep after lunch. But if anyone is interested in HLA, I could talk for days about HLA. I'm very happy to. I will keep snippets of it, but I'm not going to go into too much functional detail. But safe to say, HLA genetic association signals, when you see them, are incredibly complicated, and I still don't think we really know how to go from genetic association signal to biological plausibility or biological function. But for the purposes of this, I thought that this signal fell out into sort of two general groups. You've got these two alleles that are driving this big effect. If individuals carry a particular allele of HLA-DQB1, it's 06, they generally have a higher antibody response, whereas if individuals carry another allele, which is in DRB1, happens to be amino acid substitution, they had a lower antibody response. So if you split people into three groups, those people are carrying either sort of DQB1 or the alternate one, you can quite clearly see as you go through different time points from left to right following vaccination, that this sort of pattern is maintained. So at baseline, before vaccination, there is no difference, as you would expect, very low antibody levels in general. We had removed people who had been had evidence of prior exposure, so we do see a few people with high antibody responses. But anyway, another day. But we don't see any difference between groups. And then at day 28, which is obviously our discovery, when we do see sort of this clear pattern, and that pattern generally is maintained at the day of receiving their second vaccine and then at various days following their boost. We do get a bit of loss to follow up as you get to later time points, so that statistical evidence of difference at that later time point is lost, but you can see that there's still that pattern. So when I talk about this and people think about immune readouts, they say, well, actually, these differences in terms of antibody don't look massive. Yes, you've got quite a big statistical p-value with these associations, but what does that difference mean? And this was the lovely thing about working with the COVID trials as well. It was really the first opportunity to look not only at the immune levels, so antibody levels differing by genetics, but we could also look at risk of disease. So we, the ChAdOx trials, as with many of the early trials, were designed to try and, obviously they were efficacy trials, so they were meant to be picking up individuals that got disease. So they were individuals generally who are healthcare workers who are going to be exposed more to COVID, and so you would expect to get more episodes of COVID in the trial. So we did have quite a few episodes over time. And so we did quite a careful analysis to understand if carriage of a particular allele, and most of the work we did in terms of the functional work and understanding sort of time to event analysis was focused on this DQB1 allele. And we did find that there was a difference over time in individuals who carried DQB1-06, this allele, which is associated with an increased antibody response. So that this seemed to tell us that, yes, there was an impact on immune response at the times we measured it, and this seemed to have an effect in the direction that we would expect, i.e. greater antibody, less likelihood of disease over time. So this, to our mind, was really the first time that we could, granted in the same study, show that there was a link between immune response and genetics and also end disease, which told us that the antibody responses we're measuring are probably correlates of protection, which is what we, obviously a lot of work through the ChAdOx trials had showed already. But as with a lot of the work we've done here, we only really showed HLA signals, and that's not a massive surprise in all the work that we've done. Think about genetics and infection response. So we are trying to really ramp up this study, and just give a little flavour, and this is work we're sort of collaborating on a bit with Gus and with Nick, which is great. We always think about if you want to go big, you go with UK Biobank. UK Biobank do have a measure of antibody response against SARS-CoV-2. It's a binary readout, so it's not as powerful as that sort of quantitative responses of antibody, but we do see signals outside of HLA, and many of these look very real. We've now replicated these signals, and they make sense in terms of what they likely do. They are affecting class switching, they're affecting immunoglobulin gene usage, and they're affecting sort of B cell maturation pathways as well. So, and HLA is obviously a standout there on chromosome 6. So we do definitely see these signals. And I think when we're looking outside of the HLA, there's some of these hits which are very druggable, potentially for increasing adjuvant approaches or getting closer to universal vaccination. And we're getting very close to putting that all together. So from this first exemplar, hopefully you can begin to see that when we think about immunogenicity and effectiveness of vaccination, antibodies can hopefully be seen as being a useful proxy measure, as long as you're very careful about which antibodies you're measuring, obviously, and what you're targeting. HLA is always there, and in a sense, I've got to the point now, if I'm looking at antibody response, I'll know that the measure is looking good if there's an HLA signal there, and I sort of use that as a sort of internal positive control. But if we're going to look for signals outside of the HLA, we still need substantial power to go into this, and a lot of sample sizes, which becomes quite expensive. And certainly in the context of this post-vaccination, the signals that we're seeing, I hope, do seem to be showing something that can be intervened with and actioned on to try and help improve vaccine responses overall. So segueing into the second example, sticking with the UK Biobank theme, again, I presume UK Biobank won't need much introduction in this audience. But we were approached by UK Biobank 10 years ago now and asked for help to think about infection more broadly within UK Biobank. So working with Naomi Allen, it's now obviously chief scientist in UK Biobank, and then collaborating with Tim Wartebauer, who's based in Heidelberg in Germany. After lots of discussion with lots and lots of people, as I mentioned 10 years ago, we thought it would be a good idea to look at antibody levels against multiple infections, so now moving away from vaccines. And in fact, for the first analyses, we've made an active decision not to focus on vaccine responses and to focus on sort of infections that aren't necessarily targeted with infections, with vaccination, sorry. And so we wanted to look at a range of infections that we thought would probably be important in the risk of non-communicable disease in the UK Biobank population. So again, multiple meetings, brought lots of people together, and this was the final set of infections that we decided to look at. So we've got all of the herpes virus family members, we've got chronic viral hepatitis, so hepatitis B, hepatitis C, got retroviruses, HIV, HTLV1, human papilloma and polyoma viruses, and chlamydia, Helicobacter pylori, and Toxoplasma gondii. We decided to look at these infections because A, we thought that they would be linked with disease such as cancer or cardiovascular disease or neurodegeneration. There was some provisional evidence that they might be linked and that they could be measured using antibody responses. We then used Tim's assay, which is Luminex technology, which we'll just describe very briefly, although many of you will probably be familiar with it. It depends on conjugation of your antigen of interest. You have to choose your antigen, which is from the bug. You express that antigen. Then you can conjugate that antigen. So the antigen here is shown in a sort of yellow squiggle. You conjugate that to a fluorescently labeled bead. You then put that complex in your serum or plasma of interest. You allow any antibody in that serum or plasma to bind to that antigen. You then wash that complex away. You then incubate that with a secondary antibody, which is labeled with another fluorescent label. And then you wash that, and then you put that through a flow cytometer. And that flow cytometer has got two lasers. One is to detect the bead, which is fluorescently labeled, and the other laser detects the sort of quantity of antibody bound to that bead. And then the really nice thing about that is you can have a multitude of different beads, all different fluorescent labels, and all with a different antigen on it. We routinely do this with Tim's lab for 50 to 100 antigens, but you could go higher, and some people got up to 500 antigens if needed. But this is what we looked at. We then applied this technology, looking at all these infections, to a pilot sample of UK Biobank, so just under 10,000 individuals. And we found that the sort of estimates of prevalence were exactly what we would have expected for this age of cohort. So that was sort of reassuring in itself. We could then do various analyses using the demographic data within UK Biobank. So we could see that the expected the seroprevalence that we had measured varied widely depending on self-reported ethnicity, where individuals of a non-white self-reported ethnicity generally had higher seroprevalence estimates than those of white reported ancestry, which has been well reported in many studies. And then we could use sensitive data such as lifetime number of sexual partners, as reported in UK Biobank, and look at many of these infections, which are sexually transmitted infections. So on the left here is chlamydia trachomatis, and on the right is HPV-16. And you can see that the sort of categories of lifetime number of sexual partners, you see a significant increase in the seroprevalence here for females, but we see it in males as well. So again, although this is quite striking data, it really shows that the data, the antibody levels that we've generated are probably telling us something real about the infections we're trying to look at. Although it's this relatively small sample size, overall, it was still one of the biggest studies we've looked at with this many infections. This pilot study, actually 10,000 randomly selected individuals doesn't give you an awful large number of any endpoints to look at if we're going to try and test for association between infection exposure and a disease outcome, sort of a non-communicable disease. But here we've tried to look at CMV. So CMV has often been linked with risk of cardiovascular disease endpoints, often because we think that it might be driving some low-grade chronic inflammation and putting people at risk of cardiovascular disease. So we, as part of a group, looked at sort of the risk of cardiovascular disease and stroke over time from the point of recruitment and sampling of individuals and defining people who are seropositive or seronegative against CMV. And if we did it sort of a simple unadjusted model, so just looking at sex and age as covariates, we appeared to see an association signal. But when we were a bit more careful in terms of what we were adjusting for, in terms of deprivation index, and other sort of background demographic features, this sort of apparent association went to null. We still don't know whether we're doing the right thing by adjusting for all of these covariates. Constantly think about that. We'll think about that more as time goes on. But obviously what we as a group were mainly interested in is the genetics. So we can look at all these antibody responses. The nice thing about Luminex is they're all quantitative, or I think they are. You can sort of look at a sort of individual GWAS of each of these different antibody responses. And this is essentially what you see. So this is a circularized Manhattan plot. And what we've done is we've just sort of laid one on top of the other of all the Manhattan plots that we see. And then we've colored the peaks by the number of antibodies responses that have an association at that sort of locus. And this big yellow line here is the HLA. So we basically see at least one antibody response from every infection that we looked at has an HLA signal. So that tells us that it's probably working. But then you see a number of other peaks around the genome, some of which are specific to a specific antigen, which I think is quite interesting. But some of them do seem to be shared across multiple infections as well. And one that we particularly worked up is this peak on chromosome 4, which falls within the NF-kappa B region. We found that genetic mutation across NF-kappa B is really not well tolerated. And we know that from population studies. And if you generally have mutations in that region, you generally have sort of inherited immunodeficiency. But there does seem to be this one variant that we identified that is a more common variant that actually seems to carefully balance how much antibody you're producing. So in this bottom plot here on the right, this is basically the forest plot of the effect of this variant on antibody responses against multiple antigens as we've measured it in UK Biobank. And you can see that it sort of generally decreases the amount of antibody in those individuals. Whereas if you're looking at the same variant in multiple other studies, whether you're looking at it in UK Biobank or other studies that we're looking at, it increases the risk of people having ulcerative colitis, but decreases the risk of people having hay fever. So it's this really careful balance of immune regulation and immune response that we know is what NF-kappa B1 does. Again, this is interesting, but NF-kappa B1 is going to be so difficult to pharmacologically intervene with that there's not much we can do with that at the moment because it's going to have such a critical regulatory role. But does, I think, tell us that these genetics tell us something biologically meaningful. We can do other things. We can look at HLA genetic correlation. This is a very simplified way of doing it. We, again, just did this just to say, is the data behaving in the way that we expect it to? And we find that the effect of HLA allele carriage is in a positive direction for those individuals in terms of response against EBV antigens and also for multiple sclerosis. So this seems to suggest there's quite an exquisite link between Epstein-Barr virus and multiple sclerosis. This is not anything that, this is very topical. This just adds this weight. This has been shown before, but we clearly see that signal in the same direction. And then more recently, we've got very excited about a peak that we had initially ignored, partly because on a Manhattan plot, it fell very close to HLA, which seems silly, but just because it's in a completely different part of the genome. But it's a very real peak, falling with a specific infection, isn't seen with any other infections, and seems to link very carefully with other cancers. I've sort of been sworn to secrecy about it, but quite happy to share it here. We're really excited by it. But basically, I think it is showing pretty good evidence that an infection and an immune control of infection is probably linking to subsequent risk of a variety of cancers. And it's telling us something about immune pathways that will regulate that infection control and probably also regulate people's risk of cancer if they're exposed to that infection. So I think that could hopefully be quite exciting and just a sort of tale of things to come for the future. So for that example, hopefully more evidence that actually looking at quantitative levels of antibody can be helpful for lots of reasons, but from a genetic perspective, when you're looking at this, it does improve power, allows us to see lots of signals, both in an infection-specific way, but also in a sort of generic infection way. This is not just looking at total antibody. This is looking at infection-specific responses. And hopefully, with a bit more time, we can really begin to understand links between infection exposure and what are classed as traditional non-communicable diseases for the future. And it's great that we've got the funding now. This is the data I presented from the first 10,000, and we'll have data on the next 50,000 from the UK Biobank available next year. So that data is being generated as we speak. So now onto something quite different, sticking with the natural infection risk. I was thinking about putting this in, but then speaking to George, I thought I had to put it in. And this is going to low to middle income country settings and think about norovirus. So as many of you know, norovirus causes infectious diarrhea. I'm sure many of you have unfortunately been personally acquainted with norovirus. Working on the wards, I'm guaranteed to personally acquaint with it about once every 4 years, which is a bit unfortunate. And the reason we probably get multiple episodes of it is because there are multiple viral genotypes. So there's common ones that circulate around the globe, but then there are other ones that come out in little clusters and outbreaks. And there's some but little crossover in terms of protection against those multiple viral genotypes. It's very complicated talking about human genetic studies with norovirus, because norovirus, we talk about the different sort of families of viruses, genotypes, and then when we talk about human genetics, obviously it gets a bit complicated. It's really important in low to middle income country settings. There's a high level of morbidity and mortality associated. It's probably under-recognized because diagnostics aren't great, and we know that diarrhea causes a lot of deaths in those under-resourced settings. And what we've known for quite a long time from sort of natural infection settings is that a particular human genetic difference massively influences the risk of getting norovirus. And that's in a so-called secretor status. So secretor status is defined by your FUT2 genotype. And if you sort of carry 2 null mutations for FUT2, you are defined as a non-secretor. That means you don't express your blood group types, your A or B or O, on some cell types, and particularly you don't express that blood group on your gut cells. Whereas if you're heterozygous for sort of your wild type variant or homozygous for that wild type, you're defined as a secretor. That means you do express your ABO blood group on your surface of your cells. And that's really important for some infections, and particularly for norovirus, because norovirus uses ABO blood group as its way of entering gut cells and causing disease. So people have said if you're a secretor, you'll get norovirus, but if you're a non-secretor, you won't get norovirus. And I have to admit, when I began to look at antibody responses against norovirus, I didn't really know that work. But my sort of journey in infection genetics started in Uganda, working with this phenomenal person, Alison Elliott, on the northern shores of Lake Victoria in Entebbe, which is just south of Kampala. And Alison sort of built 2 fantastic cohorts, and particularly the Entebbe mother and baby study, which are sort of now essentially birth cohorts in Uganda. And she also looked at a sort of Lake Victoria Island study. So these are either looking at EMABs, which is centered around the mainland, which is Entebbe, and then another study, Levispa, which is looking in the islands of Lake Victoria. And through collaborators who are based in Cambridge, we used antibodies against norovirus and norovirus genotypes to understand the burden of norovirus in age-specific ways in both of these cohorts. And you can see that on the x-axis is just the sort of age. So EMABS is single longitudinal study. So these are the same individuals year on year, whereas Leviswa is more of a cross-sectional design. But you can see that the exposure rates to any type of norovirus is incredibly high by age 1, and certainly by age 2, most people have been exposed to norovirus. So it's quite a simple thing that I got asked, can we just GWAS it? I mean, that's the sort of mantra of our lab, as I'm sure it is many people here as well. But let's just GWAS it and see what it shows. And we saw pretty phenomenal effects in, we saw 2 loci, 1 locus, particularly in our Manhattan. This is of a modest sample size, quantitative antibody responses. I haven't shown the Manhattan, but basically the FUT2 locus just screamingly high. And when you look at, so these are the different viral genotypes. So you talk about it as GI1, GI2, GI3, GI4, G24. And you can clearly see that compared to our antibody responses from our COVID vaccine, they're only subtly different between groups. You've got a stonking difference here between groups. In the individuals that are secretor negative, yes, they've got quite a significant amount of variance, but those people who are secretor positive have a much greater antibody response. And that's particularly striking for this G24 genotype, but we see it for other G2 types and less so for some of the other genotypes. And initially when we looked at this, we thought, well, this doesn't make as much sense, but actually it makes perfect sense, because it's dependent. If you're not secretor, if you're a non-secretor, you don't have your blood groups on this gut cell surface. The norovirus just passes through the gut, doesn't cause any disturbance to the human physiology at all. Whereas if you are a secretor, virus can attach, virus invades, causes its disease that it likes to do. Its main purpose is to replicate and generate more of its progeny, but in doing so, it causes a lot of human tissue damage, and that's what mounts the antibody response. So people are only getting the antibody response, and at significant levels, if they're secretor positive. And then what was really nice is that we can then begin to understand, well, is there a subtle difference in terms of ABO blood groups in these individuals? So then going further in terms of molecular understanding. And although it's a bit more subtle, the sort of G12 genotype exemplifies this the most, in that antibody levels are higher in individuals who are either O blood group or A blood group, as opposed to people that generally carry a B blood group. And biological plausibility, I haven't shown the data here, but there is a true interaction. So we only see this effect if people are secretor positive. So it's really nice internal controls to give us a feeling of molecular mechanism, and going a step further than what had been shown previously, in that we know secretor status affects the risk of disease, but this tells us that ABO blood group types are specific for different genotypes. And we would love to say that that's going to give us something that we can tangibly use to reduce norovirus incidence, but we can't yet. Yeah, Gus.
Why do you see interaction at G24?
Yeah, we do. There is a reason. It's a long time since I've looked at this. The effect's not so great, and I don't think there's so much blood group specificity there. But it's true. We definitely saw the effect the greatest at the G12. Yeah, because we saw plenty of episodes, obviously, of G24. Now I'll sort of keep going with exemplars before the lessons learned. This was a study that we were looking at not just wild type infection, but also looking at vaccine responses again, but specifically in children. We were very interested to understand how genetics influences vaccine response in childhood generally, but particularly in low to middle income country settings. So again, working fantastically with Alison, but also with a range of wonderful other collaborators in South Africa and Burkina Faso. And we looked at a range of vaccines that were delivered in infancy, which are routinely given around the world. So looking at diphtheria, pertussis, which causes whooping cough, tetanus, Haemophilus influenzae, hepatitis B, and measles. And again, we looked at antibody responses as sort of readout of vaccine response at 1 year of age. And we we thought we'd done quite a good job again to 2,500 individuals, but obviously still quite modest in terms of traditional GWAS studies. And took a very long time, about 7 years, to get these results. So hopefully make people feel better if they're taking a little bit longer to get analysis done. But lo and behold, after all that time, we found associations across the class 2 HLA region. And these were for 5 vaccine antigen responses. 3 of the vaccine antigens are all for pertussis. And because we look at different antigens for pertussis, the multitude of antigens are given as part of the vaccine, and then also for diphtheria and hepatitis B surface antigen responses. And these are all genetic association signals colored by sort of linkage across the region. And you can see that the sort of patterns of association we're seeing are all quite distinct. So these are all very antigen-specific associations that we're observing. I think what is interesting is we don't see HLA associations for a few vaccines, so particularly for tetanus, measles, and HIP, which again is something that we think of quite a lot to try and understand why that is. And at this level of signal, sort of sample number, we don't see any reliable genetic association signals outside of the HLA, unfortunately. But again, probably doesn't make too much surprise now when we think about the sort of COVID vaccine responses that we've looked at. Just a little bit more on HLA. I spent a long time trying to think about HLA in this population. We performed really detailed characterization of HLA across Africa. So this is just an example of that. So we sort of typed HLA to really high resolution and multiple samples across Africa. And the 3 main ones that we looked at for GWAS were Entebbe in South Africa and Burkina Faso. But we looked at others from 1,000 genomes. And these are pie charts that sort of representing the diversity of what we call 2-digit alleles, so sort of very low resolution way of describing differences between HLA alleles. And you could just see that sort of pie chart segments for each of these alleles across all of these African populations are really quite different. And then when we compare those to Asian populations or British populations, again, just enormous diversity. I mean, we've known this for a long time, but this really showed that across the continent, we've got really quite significant diversity that's difficult to understand genetically. We worked a lot on trying to understand the impact of genetic variation on HLA gene expression. So we use lymphoblastoid cell lines to try and characterize this as looking at the impact of SNPs on expression of particular genes across the region. And we find that within African cell lines, there's a very large contribution of sort of genetic variation to HLA gene expression. And we think that probably modulates quite a lot of the effects that we're seeing with vaccine responses and HLA signals, but there's certainly more work to be done. But I think probably for this audience, this was probably one of the biggest findings that we're still noticing. Big question is, how much does genetics play a role in driving differences in antibody? And then if you believe what I'm saying, that if we look at an antibody carefully, that might tell us something about disease. We've fought decades of resistance saying, well, actually genetics compared to things like sex or BMI or deprivation status, genetics is nothing. And we've looked at it quite crudely, but no matter how we look at it, we think genetics plays really quite a significant role. And that's what this figure tries to show. So this is just the proportion of variance of our quantitative responses against antibody responses that we've looked at. And the green bars are that sort of proportion of variance from anywhere up to sort of 10 or 20% for the genetics. And this is just looking at the HLA signals that we're seeing, as opposed to other things like sex, HIV status, or sort of weight for length status. And we've looked at a number of other variables as well that were available for the African infants. And really, genetics plays quite a consistently large role. The only thing that is always larger in most studies is the time between sort of the last vaccine and measuring the antibody, which is exactly what you would expect. And we had the greatest variance in terms of that sampling time point for Burkina Faso, which explains why the bars are so big there. But I think from these two, the take-home messages are, I'm very biased because I do like looking at antibodies and I think they tell us a lot, but actually genetics plays quite a significant role in impacting antibody responses. probably a bigger impact than we've really appreciated before. And I think it can tell us things about infection susceptibility, and it can be really effective in low to middle income country settings. But so far, at the power we've been dealing with, we are finding cool things, but they're really complex regions of the genome. And working out how to clinically translate that or understand it functionally is really, really hard. It's fun, but it's hard, and takes up quite a lot of time. But it doesn't stop us doing it, and we're doing that with lots of other studies. So I just realised I've taken a like massive whistle-stop tour of loads of different studies, but hopefully stimulating a few ideas and thoughts. And my take-home thoughts from putting together this talk were, using this data, do we really think we're going to be able to prove links between infection and non-communicable disease using genetics of antibody response? I think so, but I'll see what other people think. Can we find novel druggable targets using these approaches? Again, I think so. So far, I think it's probably easier in the context of chronic viral infections. These acute diseases, so when we think about those two case exemplars I presented at the very first couple of slides, I think it probably is easier for understanding links between chronic viral infections or even acute viral infections or other sort of infectious pathogens and non-communicable disease. And unless you've got a real unique scenario such as COVID, maybe it's going to be more difficult to do with other acute diseases. And although we're using this approach, obviously, we are still trying to think about GWAS of typical infection descriptors, which I'll talk about just very briefly on the next slide. And then I know every time I talk to, certainly Nick, always challenges me on the sampling frame and thinking about, are we sure the genetic signals aren't just a measure of some other confounding or bias in the sample set, which hopefully we're getting around a little bit with measuring quantitative antibody responses. Not perfectly, because it depends on who we're measuring that in, but hopefully that's getting a little bit around that, so I hope that's helpful. But one thing I'm really struggling with, and I think we're all really thinking of a lot, is these are still dynamic measures, and we're making a big assumption in the analysis we've done to date that they're stable markers individual to individual, and we're looking at differences across individuals. But antibodies are not stable really over time, but we don't really understand how dynamic they truly are. So that's sort of an unknown for the time being, I think, but it would be fun to work out. Just very briefly, again, I should put in this is unpublished, slightly sensitive, and I'll probably be told off, but it's something we are excited about. This is trying to get at actual infection diagnoses a bit more. So this is using UK Biobank data again, and this isn't widely available, but we are allowed to sort of talk about it a bit. But we have linked UK Biobank with the so-called second generation surveillance system, which is a national sort of reporting system for microbiological isolates. And this gives us quite enormous power, really. So we can look at 58,000 isolates of Staphylococcus aureus in UK Biobank. And we can run a GWAS where we're comparing individuals who have that positive isolate to people that don't have an isolate currently reported. UK Biobank is quite mature now, so it's probably about the right time to look at it. And we do see signals come up, but they're not massive signals, even at this power. And obviously get very excited and look at some of these peaks, and actually everything that Nick and Gutty Boy told me, probably right. It's probably telling us more about obesity in the population. And other health factors rather than anything really biologically meaningful, apart from our friend in chromosome 6. So, yeah, obviously it's not going to stop us looking, but don't know, we do need to keep looking for other ways of understanding the impact of genetics on infection. So maybe looking at antibodies, well, I still think that looking at antibodies is the right way to do it. And we're trying to get the money to do it in all of UK Biobanks. So we've got the next 50,000 coming up next year, as I said. But I think there's real opportunities if we can get it into scale, really looking at every single sample in UK Biobank. I know it's a very biased cohort in lots of ways, but it's enormous in number. And if we're going to really try and understand the multiple variables that can be driving differential risk of disease and subtypes of disease, this is the cohort to do it for these types of diseases, I think, or at least to begin to get a handle on it. So that's why I'm spending most of my time at the moment, is shopping around for £20 million. So if anyone knows where we can get £20 million, that'd be great. So that's sort of the end of my talk, but it would be remiss of me not to mention a vast number of people. That's just been so fun, and I've learned so much from working in multitude of countries and settings, and just everyone just saying yes. And I realized as I was going through this yesterday, I didn't invite you guys. It's been fun working with you guys as well. But I can say yes, thank you to you guys in person. So but it has been fun, and hopefully will continue to be fun, and I hope to collaborate with some of you for a long time yet, and hopefully more of you in the future. So very happy to take any questions for the remaining of time.
I'll sort of open the chat. Oh, there is a question online from Anu, who is a paediatric ID physician. Anu, do you want to unmute and say it? Or do I? He can't unmute. I have to oh no. Oh, I didn't know about any of this. How do I unmute him?
Do it there. Yeah, just do that. You just do it.
Allow to talk. Anu, you're free.
That's all right.
Right, can you hear me?
Yeah, just about.
Okay, thanks, Alex, for a fantastic talk. Alex, I was wondering, you're measuring binding antibody using your Luminex assay, presumably to whole conserved antigen. And I suppose we're missing, therefore, a bit of the depth, both in terms of epitope specificity, but also in terms of the broader functions of antibody, considering you're measuring binding antibody, but antibodies do lots of things, both not neutralizing and non-neutralizing.
Yeah.
Is there a system serology stroke GWAS type association planned in this work stroke approach? Thanks.
Yeah.
Do you want to try and repeat that? You guys probably didn't hear that.
Yeah, sure. So the question, and great question, often get asked it. So we are looking at total IgG. This is not specifying any functional effect of particular types of antibody in terms of neutralization effect or killing ability, opsonization. We're not looking at any of that. And we're also not looking at other subtypes or isotypes of antibody, so IgA or IgM or others. So yes, you're right. And we were quite uncertain actually when we were first embarking on this, looking at the vaccine responses and then looking at the UK Biobank data, because we thought, well, actually, if that's playing a big role and we're not measuring it, we wouldn't expect to see big genetic effects. And then the other thing that we thought we might see is that we would just see sort of genetic associations that were correlated with total IgG. So there's loads of big GWAS. You know, IgG is just measured in lots of clinical studies around the world. So you can get tens of thousands. You know, the Icelandics have, I think, the biggest GWAS, which is something like 70,000 individuals that they've got total IgG level. So they're not being specific about an antigen. So we just thought we were going to see genetic association signals with sort of correlating with that. So in fact, we don't. We see very little overlap with total IgG response. And a lot of the signals that we're beginning to see, even in these modest sample sizes, you know, look functionally relevant, even without that sort of functional relevance. But you're absolutely right. It's only telling us a part of the story. You know, it'd be great to look at sort of functional levels, but unfortunately, it's just power that limits us because we don't have the throughput systems to be able to do that, as far as I know, to that sort of scale. And we really are at the moment just limited to sort of a specific antigen binding. Although it is quite exciting, you know, the newer sort of phage immunoprecipitation or VIR-scan, they claim that actually you can go down to epitope specificity, so you just express your peptides. Obviously, that depends on a single peptide rather than sort of multi-dimensional 3D epitopes that you think might be binding. But that might give us an opportunity to understand epitope-specific effects, but not functional effects, as you highlight.
Thanks, Alex. That's exactly what I was getting at. be fantastic to see those data when they're available. I mean, I guess the reason for asking is we see HLA diversity is just part of what's kept us on the planet for so long. And it would be wonderful if we could level the playing field in vaccinology by actually designing vaccines prospectively that apply to all different HLA makeups in terms of their epitope specificity, regardless of your genetic background, but also your prior exposure and original antigenic sin and those other matters.
Yeah, absolutely. That's been the crux of quite a few of my grant applications, which have been unsuccessful. I think there's, you know, I think it'd be great, and I think there's multiple ways of doing that. Either you're choosing a peptide that it's, you know, a single antigen that you think is going to be cross-reactive to multiple HLA alleles using whichever method you want to, or you just put multiple different epitopes in your vaccines that you know are probably going to drive a, you know, variable, more universal type effect. And we have to do that across multiple populations. It does seem crazy to me that we're not doing that, because there's huge fluctuation in HLA allele frequency between populations, and we often don't know the populations that need it the most, what those allele frequencies are. So yeah, couldn't agree more.
Thanks, Alex.
Cheers, Anna. Thanks. Anyone else got any questions?
Yeah, I think a very interesting talk. I was just wondering about the association you saw between, I think it was CMV and ischemic heart disease.
Yeah, ischemic heart disease is great.
So I was thinking, in the fully adjusted model, it seems like you adjust for any factor that could mediate that association. So if you know, if you want to adjust for all these factors, what pathway do you think the association would go through? Yeah,
no, absolutely. And we've begun to think a lot more about that. Seemed like the right thing to do when we first did it, but think about it more and more, and you say, well, actually, and of course, coming speaking to you guys, it's going to be something you've thought about an awful lot. And I'd love to think about more about that, because which ones, you know, which pathways do we think it is being acted through? I think irrespectively, if we're going to begin to answer that, I think we need more power for whatever we do. So I think waiting at least for the next 50,000 is the time to really start addressing that. A, because we've got more samples, and B, because we're going to have more events by then anyway. And I think that's when we can begin to experiment with different pathways and understanding, do we see any effects? We've tried to have a go with the 10,000 data set to use sort of basic graphs and different pathways, and trying to test for models that we might assume would fit more with a childhood level of exposure of CMV versus an adult level of exposure. And there's a hint that actually individuals who are younger might be more at risk of developing disease that you might expect. They've had CMV for longer, so they might have more risk of disease. But it's difficult with sample size, and also the fact we haven't measured CMV at that time point. So lots of downsides to it. But we'd love to think about that more.
Any further questions? I've got one. I'm an infection person, so I'm interested in adult infection generally, bloodstream infection, serious infection. And you know, nearly everything you present is that's not true. You know, there's a lot of viral work. There's a lot of pediatric infection. What sort of percentage, if you had to guess, of infectious agents do you think this approach could broadly be helpful for? Or do you think there's going to be a portion of antigens that simply, for whatever reason, do you have a kind of figure in?
I mean, we could run it for any antigen.
Yeah.
And I think.
But do you think it'd be useful for Staph aureus?
Yeah, well, yeah, we need to know more about it. We need to know, and people are obviously working on it. So for strep pneumo, I think we're getting there slowly in terms of understanding which antigens are probably important and which cause disease, and which the immune system really needs to mount an antibody response against. And I think once you know that, and once you know that the antibody measures that you're looking at are reliable, and that your assay is working, and you're not getting some non-specific responses, we could do it. But it's just choosing the right antigens. It's easier for viruses because there's fewer antigens. You can choose your antigens. But I think if you've got the right antigen, I think we should do it. We've done it for H. pylori. And actually, the genetic associations we're seeing with CAG, which is sort of thought of as being the main driver, look quite interesting. But we need larger samples.
Okay. Any other questions? Anything online? I don't think so. In which case,
Great. Thank you very much.
thanks so much for coming.
Thanks.
Appreciate it. Can I have another clap for Alex for coming?